Hello and welcome to a new abberior webinar. My name is Clara and I am an application specialist at abberior. Thank you for joining us today when we will explore the possibilities of single molecule tracking with nanometer precision and the MINFLUX microscopes. Therefore, today we are visiting the EMBL Imaging Center, where many MINFLUX tracking experiments have been performed. We will first hear a talk by Jonas Ries, here from the EMBL, who will explain us about kinesin tracking. And afterwards, we can see how these experiments are being performed together with Takahiro Deguchi and Christopher Heidebrecht. Both of these sessions have been pre-recorded, but we will all join together live at the end of this webinar for a live Q&A session. So if you have questions anytime during this webinar, please type them down in the chat box and we will get to them at the end of the session. At abberior, we are developing microscopes and probes for super resolution microscopy for more than 10 years. Over the time, there has been constant new development, and one of the more recent ones was the release of the first commercial MINFLUX system. If you're studying the movement of molecules in vitro or in living cells, we are living in exciting times. With MINFLUX, we now have a tool at hand to track single particles with high precision and high temporal resolution. With MINFLUX, we can achieve nanometer precision with a probing rate of up to 10 kHz. This is 100 times faster than with camera-based techniques. With MINFLUX, we can now study molecular dynamics with new spatio-temporal resolution. In MINFLUX, we are localizing single emitters one at a time by probing around the estimated molecule position with a donut-shaped excitation beam. We then gradually decrease the size of the probing pattern to further increase the localization precision. Once we define the precise molecule position, we lock onto it with the zero intensity of the excitation beam and follow it around until it no longer emits. We are joined today by Professor Jonas Ries. His group here at the EMBL has been developing technology for super resolution microscopy to study complex protein machinery in living cells. The Ries lab will soon move to the Max Perutz labs in Vienna, where they will continue to push forward super resolution microscopy for structural biology in living cells. Thanks for being here today with us, Jonas.
Yeah, thank you so much for having me here and for the invitation.
Jonas, you are a physicist by training. If you think back, what first got you excited about microscopy?
Yeah, it kind of was a long journey. So during my physics studies, I was super fascinated by the real fundamental physics question, by the universe, by the quantum world. So I started my PhD in quantum optics, experimental quantum optics. But then after a few months, I realized I was really not good at it. I was too clumsy and too impatient for these super complex setups one has there. So then I joined a biophysics group, that of Petra Schwille in Dresden, and got into contact with biophysics and microscopy. And there I developed a method, for instance, correlation spectroscopy, in this case to measure diffusion and mobility of biomolecules in membranes. And this was kind of my first gateway. But then when I was looking for a postdoc position, just the Nobel Prize, the Nobel Prize was not announced, but the technologies, the super resolution technologies had been developed. And I was super excited by those and decided to change my research and to push these technologies forward. And since then, I'm really hooked with this technology.
Cool. Mm-hmm. And now you will be moving to Vienna with your group. Mm-hmm. What are you most looking forward to at the new research site?
So I think the Vienna Biocenter, where we will be located, is a super vibrant community and science center. And we hope to find lots of collaboration partners who will use our advanced technologies and who we can help pushing the biology forward. And I'm very excited, of course, to join this community there.
Great. So thanks for coming here today. And now we are looking forward to your talk.
Yeah, thank you so much, Clara. Hello, everyone. So my group here at the EMBL in Heidelberg is very interested in understanding how complex protein machines are organized in cells. And for this, we are developing new tools for super resolution microscopy. I think many of you have heard about this resolution revolution in structural biology. with the development of the new electron microscopy technologies that now allow us to solve structures of proteins even inside cells, and that with angstrom resolution, and that's absolutely amazing. However, we also need this high resolution in order to identify specific proteins in the electron densities, and to get such a high resolution, we have to typically average over thousands of identical structures. This is now not so easy to do when we look at complex and more dynamic and less regular protein assemblies. This is why I think that optical super-resolution microscopy can be the ideal complementary technology to the structural biology techniques. Of course, we will never have the angstrom resolution of electron microscopy, so in the best case, the resolution is limited by the size of the fluorescence label what we are using. But we have the advantage that we have the high contrast of our fluorescence label, and therefore can look at individual structures without any averaging. Also, optical microscopy and super-resolution microscopy is, at least in principle, compatible with live cell measurements and can help us to investigate these protein assemblies and their dynamics in the living cell, something that, of course, electron microscopy cannot do. So the vision for my group is now to develop new super-resolution technologies that allow us to measure the structure and the dynamics of molecular machines in living cells. And the technique that we have been using so far mostly is called single molecule localization microscopy, and you might know these techniques also under the names PALM or STORM. Here we have to label our biomolecules with fluorophores that we can switch on and off, and then we acquire tens of thousands of camera frames, and in each camera frame we switch on only a very small subset of the fluorophores, because then when we have these very sparse fluorophores, we can determine their precise position, and from the positions reconstruct a super-resolution image. So this image can have a resolution 10 times and up to 50 times better than our diffraction-limited microscope. So basically, by developing these super-resolution techniques, we could overcome the long-standing resolution limit of optical microscopy that has been literally set in stone here in the gravestone of Ernst Abbe. And it was replaced now by the new super-resolution limit. Here the resolution scales now with the old resolution limit divided by the square root of the number of photons that we detect. And again, for a long time, for many years, people thought that's it. That's as good as it can get. We can never localize fluorophores better than with this new super-resolution limit. But then Stefan Hell, who already had gotten the Nobel Prize for the development of stat microscopy, had a smart idea of localizing single fluorophores even better. And this is the development of MINFLUX. So MINFLUX is a method to determine the precise position of single isolated fluorophores. And to use it for imaging, of course, one can combine it with single molecule localization microscopy and then localize the fluorophores one after the other if one has these blinking fluorophores. So what is the idea of MINFLUX? How does it work? So in MINFLUX, one combines kind of the idea of single molecule localization microscopy with ideas from stat microscopy, and that is to use a patterned beam, for instance, a donut beam that has a dark center. And by scanning now this patterned beam around the single fluorophores, we can determine the intensities, and knowing the position of the beam, we can precisely triangulate the position of the single fluorophore. And that's much better than if we had looked at the fluorophore with a camera. So why is this? Why do we get this higher precision if we use this patterned beam compared to using a camera? Well, imagine for a moment that you know where the fluorophore is and that we position our donut precisely on top of the fluorophore so that it is in the dark center. Then we won't see any emitted light, any photons. But as soon as we move the donut ever so slightly, let's say by a single nanometer, suddenly the fluorophore will see a little bit of light and we will detect photons. And we can easily distinguish no photons from a few photons from a few more photons. And this is what gives us the high precision in this MINFLUX approach. So this new precision in MINFLUX is now given by this equation here. L, that's the size of the scan pattern divided by 2 times square root of 2 number of photons for the 2D MINFLUX case. So compared to our old localization microscopy resolution limit, we gain twofold. First of all, we have these 2's in the denominator, and second, we have the scan size L instead of the size of the Poinsot function R. And L we can choose basically arbitrarily small and therefore reduce further the localization error. Now, how much can we push this? If you look at this equation here, how would you choose L to get a smallest possible sigma? Well, the answer, of course, is L equals 0, no scanning at all. Unfortunately, this is not correct. Otherwise, it would be much easier to build MINFLUX microscopes. And the reason is that, first of all, this is an approximation that does not take into account background, which can be really limiting. But the other reason is that this equation is only valid if the fluorophore is within the scan pattern. But when we start our experiment, we don't know where the fluorophore is. And therefore, we have to use a really large scan pattern. To improve on this, again, the group of Stefan Hell a few years ago developed a MINFLUX approach that kind of zooms into the single fluorophore. It starts with the large L pattern and then determines the approximate position of the fluorophore. Then it recenters the scan path on the fluorophore, reduces the L size, and after a few iterations, one can get a much better precision for the given number of photons compared to starting with a constant L size. And actually, this approach can even beat the 1 over the square root of n dependence of the localization precision, because now it scales with 1 over square root of n to the k. K is the number of iterations. So at the EMBL, we were very lucky that we received the first commercial MINFLUX instrument from the abberior company in our imaging center. And here you see an example how we could image now a single nuclear pore complex in 3D. With a down to 2 nanometer precision, and you can appreciate the high 3D resolution that we get that allows us to very easily distinguish the 2 rings of the nuclear pore complex, but also basically all the individual proteins in this complex. But MINFLUX turned out to be also quite useful for live cell imaging, and this is some data that we acquired a few years back together with the group of Stefan Hell using nuclear pore complexes where we labeled the proteins with a fluorophore that is rather dim but live cell compatible, a photoconvertible fluorescent protein MAPLE. And compared to our single molecule localization microscopy measurements, we really obtained a much better resolution with MINFLUX. Now in my group, we are trying to push MINFLUX to a technology that can take time-lapse images, that is movies of protein machines in living cells with down to nanometer resolution in 3D and a time resolution of maybe a few frames per second. But I think this is still a long way to go. On one hand, we have to make MINFLUX much faster on the technical side, and this should be possible because we are still quite far away from the physical limit. But on the other hand, we also need to find fluorophores that we can switch on and off many times with light, because at the moment we only have MINFLUX compatible fluorophores that we switch on and off once, and then we cannot switch them on again. And of course, with this, we cannot do proper time-lapse imaging because we see a protein only once in one of the movie frames. So I don't want to talk more about imaging today, but I want to talk about a second application of MINFLUX, and this is to single particle tracking. So in MINFLUX tracking, we kind of follow a single fluorophore through the cell. And we do this by determining the position of the fluorophore with MINFLUX, and then in every iteration, we recenter the scan pattern on the fluorophore. Compared to camera-based tracking, we kind of have 3 advantages. First, we have the high localization precision of MINFLUX. Second, we can get this high precision with few detected photons, which allows us to be very fast. And 3, the fluorophore spends most of its time close to the dark center of the donut, and therefore it's not easily bleached, and we get very long tracks. So we now wanted to use MINFLUX tracking to investigate the motor protein kinesin, specifically kinesin 1, that walks on microtubules, as you see here in this beautiful illustration. And it does so in this kind of hand-over-hand motion. And by doing so, it drags cargo vesicles through the cell. So in every step, the motor domain, kind of the hand or foot, takes a 16 nanometer step. And this leads to an 8 nanometer displacement of the cargo binding domain. So this motion has been studied extensively using all kinds of biophysical methods, but only on purified proteins in vitro, because we lack the spatial and temporal resolution to observe the stepping in the living cell. And this, of course, is what we wanted to change. And we did this with a big team. So we had our biological collaborators, the Kapitein group, a lot of help from people of the Ries group, other EMBL groups, of course the EMBL Imaging Center, and also engineers from the abberior company. So before we started measuring in living cells, we optimized our assay using fixed cells, and we added then purified proteins that carried the label to our cells. So the advantage is that we can now very easily control the concentration of the labeled motor proteins and tune it down to the very, very low concentrations that we need for MINFLUX. Also, it allows us to change the speed, the walking speed of the kinesin motors by changing the ATP concentration. But we started here, and this is what you see here, by saturating ATP concentration, physiological ATP concentrations. And when we then focused on a very small part of a cell and started our MINFLUX tracking, we obtained traces like this one here. So you see already that the movement is not continuous. You also see some small clusters, and these you see better if I show you here a zoom in. And already by eye, you start seeing these 8 nanometer steps of the cargo binding domain. If I now plot for you now the position versus the time, these 8 nanometer steps become very apparent, and we can start analyzing them. Now, if we wait a bit longer, we might acquire a second track close to the first one and hundreds more tracks. And when we now replace the tracks by the positions of the steps, we can further improve the precision of these tracks here. Finally, we can make use of the fact that kinesin walks towards the plus end of the microtubules and assign a directionality to the microtubules. And this is color-coded here. If we now compare this now MINFLUX tracking image with a confocal image, we do have a much higher resolution and get much more information out of it. Then we collaborated with a group of with a node group here at the EMBL to look at the organization of microtubules in neurons, and these are examples here where we looked at different parts of the neuron, and in these dendrites, we found that most of them have microtubules that run in both directions. Some of them are clustered together, and here is one part where we only have unidirectional microtubules. Now we can look more precisely into individual tracks. Here, if you look at the blue track, you see a sidestepping event, probably between adjacent protofilaments, but we cannot exclude that this also happens between adjacent microtubules in a bundle. Quite often, we observe the sidestepping after long stalling events, as you see here in the green track, and we interpret this as roadblocks, maybe even fixative, that kind of attaches proteins to the microtubule so that the kinesin cannot pass them, and it can only circumvent these roadblocks by sidestepping. To further investigate really the dynamics of our motor proteins, we reduced the ATP concentrations by about 50-fold, and now just by bare eyes you can beautifully see these 8 nanometer steps. If we now want to determine the experimental localization precision, we can look at the standard deviation of the position measurements in each of these steps, and we found a value of around 3 nanometers. But because we can measure hundreds to thousands of localizations in every single step, we can determine the position of the step with a much higher precision, with a standard error of a mean of better than 1 angstrom. But of course, in the end, that's not the resolution that we get in our MINFLUX, because we're limited by instabilities, let's say, of the sample, of the microtubule still moving, of the microscope, and also of the size of the label that we are using. But still, I think this shows that MINFLUX can be quite useful technology to map out, on one hand, the kinetics and the conformational changes of the kinesin molecule, but also the organization of microtubules in fixed cells with down to protofilament resolution. So now I would like to turn to living cells. And to do MINFLUX tracking in living cells, we expressed kinesin 1 with a halotech now at its motor domain and labeled it with a live cell compatible dye. And again, by focusing onto a really small part of the cell, we obtained many tracks. Here you see one of the tracks that we rendered as a super resolution image, and you clearly see these clusters of localizations with a distance of 60 nanometers. Again, in the movie, you can appreciate these 60 nanometer steps, and the spatial resolution we obtained was 2 nanometers, combined with a temporal resolution of better than 1 millisecond. And that's something really good compared to what one can achieve with camera-based tracking on single fluorophores. Now we can again analyze the position versus time plot and extract the length of each step and also the distance that the motor domain takes. And we can do this in hundreds of tracks to kind of for the first time extract the precise distribution of step sizes and dwell times of kinesin molecules in the living cell. So I think that this shows also that MINFLUX can be quite useful technique to study the details of motor proteins and other biomolecules on the nanometer scale in living cells. So all the data I've shown you so far has been acquired in 2D, but of course life is three-dimensional. All these movements of motor proteins and other molecules happen in 3D, and that's why we wanted to extend also the live cell tracking to the third dimension. So as you might know, MINFLUX has been used in 3D for imaging, but not yet for tracking. So we extended it by now using a so-called 3D donut. You can think of it as a kind of sphere of light with a dark center in the middle that we position at 7 positions around the single fluorophore. And by doing so, we can then determine the X position, the Y position, and usually the Z position of the single fluorophore. Compared to the two-dimensional tracking, we lose a bit in temporal resolution and spatial resolution. But as you see here, maybe I play it again, we can easily record still the 60 nanometer steps of the kinesin molecule in 3D in the living cell. We also used the 3D tracking for motor paint, and that you see here, where we can now map out the microtubules in fixed cells with down to profilament resolution in 3D. So I think this is very exciting, but looking only in one color at proteins is still quite limited, because it's very difficult to distinguish internal motion, kind of conformational changes of the protein, from external motion, how they move in the cell. This is why we are at the moment working very hard to extend MINFLUX tracking from a single color to 2 colors, because this would allow us to put, let's say, a red and the green fluorophore on our protein and then observe the distance in 3D with nanometer precision, and at some point hopefully down to 100 microsecond temporal resolution. And with this, we should be able to observe how proteins undergo conformational changes during their function in living cells. So with this, I'm at the end. I want to thank everybody involved in the projects I've been talking about. Of course, the wonderful collaborators we had for this project, my awesome group at the EMBL, and the alumni who contributed, and of course, you for listening. Thank you.
Thank you, Jonas, for this exciting insight into your research. Now remember, if you have questions, please write them down in the chat box below, and we can get to them at the end of the session in our live Q&A. So now that we have seen what kinesin tracking looks like with MINFLUX, we want to see how that actually works at the MINFLUX microscope. For that, we now join 2 members of the Ries group who will show us a MINFLUX experiment. We are now with Takahiro Deguchi, a postdoc from the lab, and Christopher Heidebrecht, a master student. So, what sample did you bring for us today?
So today we brought a human cancer cell sample with a specific preparation called the motor paint. Here we put a Drosophila kinesin labeled with a fluorescent tag, and then we expect kinesin working on microtubules.
Wonderful. Then let's take a look at the microscope.
All right, so Takahiro is now using the ocular to find a fitting cell for our kinesin tracking experiments. Our cells are transfected with GFP tubulin, so he tries to find a nice spread out cells with good microtubule filaments. Okay, do you see something?
Yep. Very nice cell, very good sample. Okay.
All right, now he found the cell. Now we're switching over to confocal mode on the MINFLUX. Yes, now we start scanning in confocal mode over the cell, and we see we lost the focus a little bit, so we have to fine tune it again to find the microtubules. Yeah, and as you can see the nice microtubules. You can kind of make out the nucleus. Yes, so this is a large field of view of the cell. So as a next step, we are scanning over a smaller field of view to get a nice position where we can track kinesin. And these white dots in this field of view are already kinesin that are either floating around or just kinesin that are in confocal mode. All right, and now Takahiro is scanning around to see if there's any moving dots that are moving in a specific trajectory. And we can see one in the corner right here, which could be a potential kinesin. So we know at this region there are walking kinesins, and we can set it as a MINFLUX ROI, and we can start the MINFLUX tracking experiment. So yeah, now this window pops up where we have our kinesin tracking experiment, and now we are hoping to start seeing tracks. Yeah, and now we just have to wait and see what happens. So, and now we already see we have a very nice track. Well, it's a rather short one, but still okay. Yeah, these points with these blue points with high localization count are kind of what we're looking for.
Oh yes.
There's another long one. Oh, it's nice.
Oh, yeah.
Yeah, and this, you can see we have these distant blue points, which are likely kinesin steps where the kinesin pauses for a while, and we get more localizations.
Okay, we can change the field of view.
Yeah, and so now we don't see any kinesin tracks anymore, so it's likely that we just don't have a, I mean, we got some tracks, but we are trying a new position to find, to see if we get some more tracks. So we're back in the confocal scanning mode. You stopped the acquisition, right?
Yeah.
Yeah. So we stopped the MINFLUX acquisition. We are back in the confocal mode, and we are now trying to go for another region. Are you trying on the same cell, or are you going to a different cell? So we are remaining on the same cell, but we are just trying a different part of the cell to see if there's any more kinesins, or yeah. Yeah.
Yeah, here.
Yeah. So again, see a few walking ones already in confocal mode because they go in a straight trajectory. So we chose this as a field of view, and we start the next MINFLUX acquisition, and we already see a pretty nice track here. That looks good, right? Yeah.
Yeah, very good. Oh.
Yeah, that looks. We see these really distinct localization clouds, which means we see clear kinesin steps. That's actually a very nice track we got there. Oh, there's another one. Oh, yeah. This one also looks very good. That's nice. So yeah, changing the region after a while of MINFLUX acquisition usually helps out to find more tracks. All right, so with this long, nice track that we've got, you see these very point-like localization clouds where we expect, or we know that kinesin takes a step because we have an N-terminally labeled kinesin. So at the head domain is the one that takes the step. So it's at the microtubule, we expect a 60 nanometer displacement. And so these point clouds should be all 16 nanometers apart. If we have a look here, Takahiro's now checking for the distances, and yeah, they should all be around 60 nanometers. These blue dots you can see here are just points where where kinesin was just stopping for a longer time, and we got way more localizations out. Yeah, is it 60 nanometer? Yeah, it's around 60 nanometers. Yes. But these are not side steps here, right? Or could that be
These
side
could be side steps.
These also could be side steps, so where kinesin steps from one protofilament to the protofilament next to it. We've also seen it in our data from the publication. So we are not sure yet, but this kind of looks suspicious for sidestepping. All right, so now we stopped the MINFLUX acquisition, and Takahiro will load the dataset in PowerView to get a different visualization. Okay, now we get a different rendering of all our tracks, and you can see it's a very colorful map. each track that the MINFLUX acquired gets a new track ID. So, and this is displayed with a different color. So you can see our very nice track that we got is this yellow one right here. Takahiro is going to zoom in on it a little bit. And what is now nice to see is that each of these points is one MINFLUX localization. And so these Takahiro is lowering or making these points smaller a little bit. And now we can also see the line along the tracks. So the power view basically connects these lines in between localizations, and we can see the track even a little bit better. That's also how we visualized our tracks in our data for the publication. Yes, and we can again see these large point clouds where there has been a lot of localizations, where there might have been sidestepping. It's very hard to tell, but there seems to be a displacement right here from here to here. Yeah, which could again be sidestepping, but we would have to look at it, analyze it further to be sure.
So we showed how we acquire kinesin tracking data using MINFLUX. Now we show how we analyze the data. For the data analysis, we use a specific software package called SMAP, and this is something we develop in our group. So I just loaded the data on the SMAP, and you can see the overview image of our entire field of view in this window. Here we see an entire window with a different zoom. For example, we can zoom into a nice kinesin track here. So when we change contrast, we can see clear accumulation of localizations in a periodic manner. Then we go to ROI Manager, and when we open ROI Manager window, we can see a low resolution image here, and if we choose one of the ROI on this image, we can start seeing individual kinesin tracks. So now we want to analyze this kinesin track, and it looks like this. So for example, if we look at in this window, this is an XY rendering image of localizations. And you can see these stepwise, let's say, accumulations of localizations. Then we can see stepwise movement of kinesin in this time versus position plot. And you can clearly see that the step size is close to 16 nanometer. Now we can analyze different things on this ROI Manager. For example, here you can see the time that we needed to make one localization. So this is exactly the time resolution of the system. And the median time resolution is 1.8 millisecond, and the minimum was 0.9 millisecond. So we can localize the kinesin position about 1 millisecond of temporal resolution. And for example, if we go in this window, that you can see that kinesin walks along one direction. So this is a time versus y position, so off-axis position. So it's orthogonal direction relative to the kinesin walking direction. And if you look at this area, for example, where I zoom in, you can clearly see that the kinesin position moves more than 6 nanometer along the y direction, the off-axis direction. And we consider this as a sidestep. So this actually is one of these localization accumulated position. So you can see the kinesin was walking along this direction, and then after staying longer time in this position, then it moved to the off-axis direction, and then continued walking along microtubule again. And since we see this offset movement, so that we consider this as a sidestep motion. And we have seen it several positions. For example, here as well, you can see the kinesin steps is found here, while the kinesin step continuing on this at this position in y, which is minus 18. So this, compared to this, about minus 10 nanometer in y position, you can see that the kinesin made a movement along y direction, which means that this is a sidestep. And we can also see that what was the signal detection rate, what is the center frequency ratio, and also how many photons we collected. And this is directly correlated to the resolution of the final image. So that this is something that we also check in this analysis. So today we have successfully acquired kinesin working on microtubules in human fixed cells, and we have also seen clear kinesin working steps. In addition, we also saw side steps of kinesin.
So thank you, Takahiro and Chris, for showing us your MINFLUX tracking experiment. With this, we're at the end of this part of the webinar. So we will now move to our live Q&A session. So if you have questions for Takahiro and Chris, please also write them down in the chat box and we will come to them now. So thanks for joining live now. And we are already getting in the first questions. Maybe as a first question for you, after this presentation and after we've seen now the experiment in progress, one question would be, does the speed of kinesin move in cells, and how could you modulate that in cells?
Should I answer that? Yeah, sure. So in vitro, we of course can change the ATP, the kinesin speed by the ATP concentration. So by lowering the ATP concentration, we can lower the speed of kinesin, and therefore it makes it a lot easier to track. Of course, in live cells, that's way more challenging. We cannot just change the physiological ATP concentrations.
Okay, thank you. So then there is a question, what's the limiting factor for improving the temporal resolution? Is that the same for other other microscopy techniques?
So this depends mainly on the emission rate of the fluorophore and also the precision. So it's a trade-off between the precision, so resolution, and the time resolution, spatial resolution are the trade-off. So if you can compromise the resolution in spatially, then you can gain in time resolution. And also, it also depends on the background, as if we can decrease the scanning pattern size, L, then we can gain in time resolution as well, because we can reduce the number of photons we collect. So these are all, optimization, let's say, problem.
Okay. So the next questions, what are the considerations of dye choices when labeling the protein?
Do you want to answer that, or I can? Sure. Okay. The consideration for the dye choices are, of course, brightness mainly. So we want to use dyes that are as bright as possible. Fluorogenicity is very important, is relatively important, so that the dye only is bright when it attaches to the ligand, or to the tag, sorry. And of course, for live cell permeability is a very important factor, that we get enough dye into the cell. Yeah.
Okay. Then there's another rather technical question. Do you use drift correction for your experiments in live cells or in fixed cells?
I can answer this. So The drift correction algorithms that we use for single molecule localization microscopy and post-processing don't really work for MINFLUX very well because they require many localizations to be measured at the same time. But the abberior MINFLUX has an online real-time drift correction built in that we use for imaging. So when we take time-lapse images, which take minutes, then we have to stabilize the sample position with respect to the objective. For tracking, correct me if I'm wrong, but usually the drifts are so small in these 10-second windows that we don't necessarily need it. But I think for the MINFLUX motor paint, we have to switch it on in order to not have drifts from the microtubules.
Yeah. And also in live cells, microtubules are moving, they are dynamic, so that we don't use drift correction because they are always changing their positions.
Okay. So then there's another, how can you quickly scan through the stack after you acquired a confocal ROI to see the particles moving?
Okay, I can answer. So we are not catching the kinesin which are working on the confocal mode, because probably they are going on, when we start MINFLUX measurements. But we know that the microtubules are good for kinesins to work in this region. That's why we choose this region as the MINFLUX ROI.
Okay. So let me quickly check whether there's a lot of questions, but that doesn't mean that you shouldn't stop writing the questions, because we will get to the questions also after the live Q&A. After some point, we will stop the session, but we will get to all of the questions afterwards also per email. So don't worry, your questions will be answered. So keep typing them, and I quickly browse through the questions to find some that we can answer now. For kinesin tracking, are you tracking only one floor for over such many frames?
You want to answer? So, I can. Of course, how we label, it could in principle be possible that we have two floors attached to each of the halo tags that we have there. However, that's quite unlikely because we use much less dye than we have halo tag there. And also, we would detect it in the measurement itself, in the tracks, by the signal, by the contrast, and by the bleaching at some point that still gives us some signal left. So overall, the majority of tracks are really a single flow that we track.
Okay. Okay, so this has been already quite some questions that we answered. I think with this, we're reaching the end of our live Q&A session, and all the remaining questions, many of them are along the same lines. We will now answer per email, so stay tuned for that. And if you have questions coming up now, not just after the discussion now, please also keep writing them. We will leave the room open for a little bit longer, so you can type your questions. So, and with this, we are at the end. Thank you, Jonas, Takahiro, and Chris for joining this for this webinar, and for preparing this webinar with us. It was a pleasure. And thank you all of the audience for joining today. Thanks so much. And goodbye.
Goodbye. Goodbye.